we'll start with Ru-Rong from Duke, who's going to be talking on omega-3 fatty acids for the treatment of spinal cord and traumatic brain injuries. Ru-Rong.
Thank you very much, Richard, for the introduction and also for organizing this session. I would like also to thank Larry for organizing this fantastic symposium. I have learned a lot now, but unfortunately I have to leave early for my daughter's graduation tomorrow. And my talk here is, as you can see, that omega-3 fatty acids and to block and inflammatory pain and neuropathic pain. Also, you can see I have a subtitle and to discuss and these specialized pro-resolving mediators, SPM, derived from omega-3 fatty acids. So here is my disclosure. So there is no commercial support for my talk. So I'm supported by NIH and DOD. So from the previous two speakers, so you have pretty good background about pain, and in the past 2 decades, the field has seen tremendous progress in terms of molecular mechanisms, right? The transducers of pain, and also the neural circuits that can mediate different types of pain, including mechanical pain, thermal pain, spontaneous pain, and also the emotional component of pain. And you can also see that chronic pain is a big health concern in this country, affecting over 100 million Americans, and it's an annual cost over $600 billion. And also you heard from the previous speaker, so that chronic pain has comorbidity with depression, anxiety, insomnia. And also there are 2 ongoing crises in this country. One is chronic pain epidemic associated with opioid use disorder. Actually, this is a good time to do pain research because NIH has this pain and heal initiative, right, to address these 2 crises at the same time. A lot of investment, over $1 billion, used to develop novel pain therapeutics. Probably this is not enough, as you know, to develop a drug, and is very, very expensive, right? So we still need an industrial support, right, to develop novel pain therapeutics. So, I want to focus on my talk on inflammation and pain, because omega-3 fatty acids and SPMs, they're one major mechanism to treat pain is to tackle inflammation. So inflammation is familiar to you, right? If you have infection and a tissue injury, you will get inflammation. Can be measured in your blood samples. This is systemic inflammation. And also I want to mention 2 different types of inflammation. One is called neurogenic inflammation. This is very specific, if you have an activation of sensory neurons, especially in the nociceptive neurons by capsaicin, right? So you will develop edema. So this is a neurogenic inflammation. And another type of inflammation I will also emphasize is neuroinflammation. So this is inflammation in the nervous system. And I will give a further introduction in this slide. So this is inflammation in the peripheral and central nervous system. A few features: infiltration of immune cells, and also activation of glial cells, including microglia cells, astrocytes, and satellite glial cells. And as a result, there is a production of immune and glial mediators, So these cytokines and chemokines, now we know not only just produced in the periphery, but also in your brain, in the spinal cord, that can drive chronic pain. Actually, we know that neuroinflammation associated with many neurological and psychiatric diseases, right? Including AD, PD, multiple sclerosis, and depression. So compared to inflammation, which is rapid and very robust, neuroinflammation can be a low degree of inflammation, but more persistent. So therefore, it's more important for the maintenance of chronic pain. So here now, okay, let me clear this one. Okay. Sorry, I think I probably this. Okay. So I want to discuss inflammation, and there are 5, let's say, cardinal features of inflammation, and pain is one of them. This is also our daily experience, right? If you have inflammation, tissue injury, you will have pain. So pain is always associated with inflammation. And we now know because during inflammation, there is infiltration of immune cells, right? Immune cells. And these immune cells will produce inflammatory mediators. And there are different types of inflammatory mediators, right? So you can have pro-inflammatory, and like prostaglandin, which is a lipid, right? And you have pro-inflammatory cytokines and chemokines, such as interleukin-1 beta, TNF-alpha, chemokine CCL2. Okay. So these pro-inflammatory mediators will activate and their receptors, right? So there are different receptors on nociceptor. Nociceptor is specialized sensory neurons, right, that can sense pain. And now we know, I think, there are receptors, right? So you have a receptor for prostaglandin, for cytokines, and for chemokines. And in addition, we know, that inflammatory mediators can include nucleotides, such as ATP. Now we also know, that in RNAs, small and like microRNAs, right? They may also serve as inflammatory mediators. And also their receptors, such as toll-like receptor, can also be expressed by primary sensory neurons. And therefore, microRNA can directly signal to sensory neurons to induce pain. In addition to pro-inflammatory mediators, which are typically pro-nociceptive, we also have an anti-inflammatory mediators, because the goal of our acute inflammation is to resolve, right, to heal. So during this process, there is production of anti-inflammatory cytokines, such as interleukin-10, TGF-beta. But today, I want to highlight the third category. That's SPMs derived from omega-3 and fatty acids, right? So they can be anti-inflammatory and pro-resolution, and also anti-nociceptive. So, and we know, that the primary sensory neurons, as you heard from the previous speakers, so these neurons can transduce pain to the central nervous system. Okay. And a milestone in our field is that two prominent pain scientists, right, David Julius and Auden Pat-Pushin, and received a Nobel Prize and for their discovery of two important pain sensors. One is capsaicin receptor, TRPV1. Another is mechanosensor, which is also important for mechanical pain, expressing sensory neurons called piezo2. And we know that during inflammation, and these two pain sensors can be activated. So here you can see also a lot of progress in the field has been made in terms of cell biology of pain. And of course, we have been focused on these nociceptive neurons, right, which has two axons, one to the peripheral and the joint and the skin, one to the spinal cord. But now we also appreciate, and that this nociceptive neuron actually is surrounded by many different types of non-neuronal cells, right? And including the glial cells, such as microglia and astrocytes, and also the immune cells. Actually, these non-neuronal cells and play a very important role and through neuroimmune activation. And Dr. Linda Watkins here at the CU and Boulder, she is a pioneer in this area. So in here you can see that these microglia cells, after nerve injury, a condition that can trigger neuropathic pain is very active, right? And upon activation, they can produce a lot of pro-inflammatory mediators, including pro-inflammatory cytokines, interleukin-1 beta, TNF alpha. That can drive chronic pain. And we have made a lot of progress, and in the, for example, central nervous system, such as spinal cord, the neuroimmune interactions actually can facilitate and prolong the development of chronic pain, right? Through neuroimmune and neuroglia, these interactions. Basically, these neuroimmune interactions may serve as amplifier, right? And of the pain signal. Okay, so yeah, and in the next session I move to a fish oil, as Richard mentioned, right? So there are many brands now of fish oil now, and they have multiple benefits, but one major benefit is for the controlling inflammation and the pain relief. So there are two major components, the EPA and the DHA. And as you can see now that during our life, we have persistent requirement for EPA. But for DHA, somehow we have more requirement in our early and the late stages of our life. And also DHA may be more important for the protection of the nervous system. So, we know that and the DHA, fish oil, DHA and the EPA can have direct effect and beneficial effects maybe through their own receptor, such as in the GPR, GPCR, and 40. So they can have an anti-inflammatory and analgesic effect. But relatively, these effects are weak. And the next I will tell you that these SPMs are derived from fish oil, and they can have an anti-inflammation, pro-resolution, and analgesic effect. And we believe their effects are more potent. And recently I did a search, actually and these SPM-related products are also on sale now. And you can, if you search, I think, and you can see that and these products actually can sell at a much higher price, right? 5 or 10 times. A fish oil, you get $10, $20, $30. But these SPM contained fish oil can sell over $100, right? So this is a very, very good business. There is no FDA control now. And because these are metabolic products, right? Any fish oil will contain certain amount of SPM. The issue is how much. Whether that amount can reach therapeutic, we don't know, right? So we don't know. But at least I'm glad that somehow in my research can be, and potentially someday useful, right? For if not really, I think people think SPM can help control inflammation and pain. Okay, so this is in a biosynthetic pathway. So EPA and the DHA, you can get and several classes of SPMs, including resolving, right, and protecting and releasing. So resolving, you have E series derived from EPA, and the D series derived from DHA. So here I think I want to show you the comparison between the analgesic and the efficacy of fish oil, right? DHA and the EPA, and also resolving. Okay. So if we use this model CFA-induced inflammation, and you can see the analgesic effect, there is a significant difference, right? EPA and the DHA are effective through this local intrathecal injection that can directly target the spinal cord. And as you can see, if you use a high dose, 10 microgram, so you can have analgesic effect. But resolving E1 as a dose is 1,000 times lower, right? 10 nanogram. So there is a difference. And also here we use another inflammatory pain model. And on the right, we compare the dose response curve between resolving E1 and morphine, and also anti-inflammatory treatment COX-2 inhibitor. And as you can see, the effective dose of resolving E1 is 100 times lower than morphine. And as we know, because these SPMs have an anti-inflammatory effect, right? So therefore, they can inhibit inflammatory mediators, as you expected. They can also inhibit activation of microglia cells in the spinal cord, because these are kind of macrophage-like immune cells in the central nervous system. But what surprising is that we found that resolving E1 can directly act on sensory neurons, right? And I told you, right, that the capsaicin receptor TRPV1 is very important for inflammatory pain and neuropathic pain. And if we do patch clamp recording and add capsaicin, you can get this current, right? Okay. But if you add a resolving E1, basically you can block this current. So this is kind of surprising, and that resolving has direct effect on sensory neuron, can produce very rapid analgesia within a minute. They can also modulate synaptic transmission in the spinal cord. But as we heard a lot, right, so that animal studies may not translate to clinical studies. And here we also tested primary sensory neurons from human, right? If we dissociate these human neurons, we add resolving, we can also block capsaicin-induced current. So there is clinical relevance. So here I think you can see there is another comparison, right, between resolving and its precursor DHA and the EPA in terms of the inhibition of ion channel TRPV1. Resolving can inhibit these channels at a concentration of 1 nanomolar. When you use DHA EPA, if you increase your concentration to 1 micromolar and 10 micromolar, and you will also inhibit these channel activity. Now we still don't know the exact mechanism, because it's in that lipid raft, because these are lipids, right? In the lipid raft, you have a lot of ion channels. And if you have a high concentration, you may disrupt that interaction. That can have an analgesic effect too. So here I think we tested neuropathic pain. So typically, neuropathic pain is produced by nerve injury, right? So for example, ligation of the sciatic nerve. And we found that actually DHA is effective if right at the time of your injury, you put the DHA next to the sciatic nerve at a high concentration, right? And it is effective to prevent the development of neuropathic pain. And also an MPD1 derived from DHA is also effective. But what the difference we found is the late stage, right? Because DHA and resolving are effective at the induction stage when you have acute inflammation, right? And when I think that resolution process happens, so DHA is effective. But if we wait for a few weeks, let neuropathic pain develop, this is typically you treat the patients, right? Because they have pain, they come to you, right? So in that case, we know that neuroprotecting MPD1, right, is effective. but DHA is not effective, even though you use a dose 1,000 times higher. We also have a comparison with gabapentin, a typical treatment for neuropathic pain. So you can see that MPD-1 dose can be 500 times lower. So, and this is a summary from a recent review article I published. Basically, these SPMs have been tested in various animal models, right? Including inflammatory pain, neuropathic pain, cancer pain, and also in animal models of skin disease such as acute and chronic itch. And we now know that and these SPMs act through G protein coupled receptors, and these are orphan receptors. Several receptors have been identified, and basically these receptors are expressed by macrophages. Activation of these receptors and in macrophages and will cause phagocytosis, which is a very important step for the resolution of inflammation. And activation of these receptors also will change the macrophage and phenotype, right? So that can have a pro-resolution. In addition, these receptors also expressed by microglia, right, which is kind of macrophage-like cells in the brain, and also neutrophils, because that activation of this receptor can control and the neutrophil activity to basically control inflammation. And recently we identified an orphan receptor called GPR37. So this can serve as a receptor for neuroprotectin D1. Okay, so we can see there is a calcium signal after activation of this receptor. And importantly, you can see this phagocytosis activity, right? If you add this one, and then they eat a lot, macrophage can eat much more. And this effect is basically disrupted in GPR37 knockout mice. And also we demonstrate that this receptor is important for the resolution of inflammation and the resolution of inflammatory pain. Lack of this receptor, and you can see that the pain, inflammatory pain does not recover. And we did some simulation, and of course there are different therapeutic strategies, right? One is you can use these SPMs, right? If you can, because the limitation is they are very expensive to produce. There are many synthetic steps, right, to make them, and also they are very unstable, right? So one strategy is to make a different delivery and a strategy. For example, emulsion to make them more stable, or for the local delivery. Another thing is if we know know-how, if we know the receptor, right, we can also develop a small molecule agonist, right, to tackle these receptors. For example, here we identified a possible ligand for GPR37 is an artesunate. This is a known anti-malaria drug, right? Because anti-malaria drug is typically think to kill parasites. But now we provide an immunotherapy for this anti-malaria drug. It may activate all macrophages. The macrophages can basically take over parasites. So this is just another way for the resolution of inflammation. Because of time and the limit, so I will not, So in the next speak, Adina will talk about traumatic brain injury and the TBI. And here you can see that in a mouse model of TBI, and the MPD-1 can prevent the development of neuropathic pain. And I mentioned that the GPR37, the receptor, is expressed by macrophages. But in the brain, this receptor is highly expressed by oligodendrocytes. And these glial cells make myelin, right? And as you can see in the TBI model, right, there is dramatic demyelination, loss of myelin, right, because we know myelination is important in associated with aging, right? Basically, increased myelination may also improve our intelligence, right? So as you can see in this TBI model, there is a very dramatic downregulation, but the treatment then can reverse this demyelination. And also, and we found this treatment actually can bring, because the receptor is expressed by oligodendrocytes, right? The treatment actually can reverse the downregulation of this receptor. I'm discussing this GPR37, right, as a receptor for neuroprotectin D1. And recently, we identified another receptor called the GPR37L1. It's also alpha receptor, which is very similar to GPR37L1. And this and it can be a receptor for another SPM called Maricin-1, MAR1. There could be multiple, let's say, receptors for each SPM. So, and this receptor, interestingly, is expressed by astrocytes in the spinal cord. And they all express in macrophages, right? They can control inflammation. But in the brain, they show complementary expression patterns. For example, one expressed by oligodendrocytes, another by astrocytes. We know these glial cells can be protective in terms of brain injury, neurological diseases. So we also identify a polymorphism, a mutation of this gene. And the loss of function of this gene is associated with widespread pain, development of chronic pain. So basically here we identify these mediators, SPMs, and also their receptors. And these receptors probably are protective, and the loss of function will lead to chronic clonification and also failed resolution. Because we think a lot of chronic diseases are associated with our failure of resolution, right? When you are young, when you have acute injury, typically you can resolve, you can recover. But in the late stage chronic disease, or when we are getting old, we lose the capacity of these pro-resolution mechanisms. And so because of time issue, so I will just move on. And GPR37L1 actually we found is also expressed in satellite glial cells. These cells, the primary sensory neurons, each primary sensory neuron is surrounded by these satellite glial cells, and they provide a nutrition, right, support to their sensory neurons. And we found through RNA sequencing that these cells, satellite glial cells, are very important for lipid signaling. And a lot of their marker genes are related to lipid signaling, right? So including the lipid binding proteins and the lipid receptor. And we think, and these cells probably particularly important to protect sensory neurons and through lipid signaling, and also through SPM signaling. So, yeah, so I have 1.5 minutes left. So I will make a summary so that these SPMs were discovered by Dr. Charlie Serhan at Harvard Medical School. And I mentioned that fish oil, DHA, and EPA and do have analgesic effect, anti-inflammation effect, especially at the acute phase, right? Acute inflammation. Presumably, because during the acute inflammation, they can be converted partially into SPMs. Whereas in the late phase, basically this conversion mechanism is disrupted. So even though you give a lot of fish oil, you may have very limited benefits. So here I think I also want to discuss a few alternative approaches, right? So as I mentioned, so if we figure out receptors, we can target a receptor, we can develop a novel method of delivery. Also, I want to raise your attention is that neuromodulation, right? Because the first speaker mentioned about autonomic dysfunction, and which can be treated by vagus nerve stimulation, right, or auricular stimulation. And we found that auricular stimulation can actually promote the conversion, the production of SPM. Other neuromodulation, such as spinal cord stimulation, a common treatment, right, for chronic pain, can also increase in the production of SPM. In addition, and that we will have a talk about a young blood or that blood product conditioned serum, right? If you condition your serum, it can also through coagulation can also produce SPM. So you can have boost your endogenous production of SPMs. So this is another approach. So I will finally, I just acknowledge my group and my collaborator in Charlie Serhan and Harvard Medical School. Thank you for your attention.
Thank you, Ru-Rong. And so we've seen the field of omega-3s opening more and more widely. Some questions or comments, please. There we go, number one.
Thank you very much for the presentation. My uncle many years ago in the 60s tried to do a venture with fish oil and converted LST, a Navy vessel, and he unfortunately ended up losing his shirt. But my question is that I'm vegan, so I use the flaxseed oil to get my omega-3s, my linolenic acids, and so it's just a real generic, 101 type question. And so that's just as good as the fish oils. I'm assuming or no, the fish oil has an ingredient. I know oil is supposed to be exceptional in that category of the omega-3, 6, and 9s. So I was just wondering, with the linoleic acid, is the flaxseed oil comparable, or am I missing out? Thank you.
You want me to answer?
Yeah. So I know, so this is in a very large area, and I think many questions remain unknown, right? Because these are natural products, right? You don't need FDA approval, right? So basically, you can sell them, and I would say there is not very convincing clinical trial, not so many convincing clinical trials, right? And to show the efficacy. Also, as I discussed with Adina, right? So you have a mixture of many things, right? It's difficult to nail down which product is active, right? Because when you eat many things, so we don't know. So, yeah.
Studies showing that if you eat flaxseed oil, which contains the alpha-linolenic acid, which is the precursor of EPA and DHA, that the effects are not nearly as beneficial as EPA and DHA. So the flaxseed oil has very low conversion to the bioactive EPA and DHA. I think Larry has a question.
So I have an incredibly simple question. And we've never had a chance to ask this of someone like you. You looked at your last slide, the second to the last slide, not the real slide, and everyone in this room actually wanted to know the answer to one question. Every morning, which supplement do you take?
Or evening.
I do take fish oil, and recently I went to a meeting in Boston, Harvard Medical School, right, organized by Charlie Serhan, right, and there are also companies selling in SPM. Recently, my wife had a low back pain, so I asked her to take, but I don't know, because we don't know how much SPM and what is a level therapeutic level you can get, right? Because there is approved and only one, to my knowledge, in the EPA, right, for treating cardiovascular disease, but that does a few grams. That's far above, what we take, right? Because you need to take 10, salmon dishes, right, every day, right? Because that probably not maybe in a preventive, more. In that case, you need less, right? But for a treatment, I would say you need to have a much high dose, to reach, especially to the right place. You need much high dose. Yeah,
Where
please.
was-
Thank you. I saw a couple slides there, and it looked like there was an injection being done. What I remember you said was we might do an injection for an acute phase inflammation, and it looked like that was being injected into the actual area where the inflammation was, rather than an IV that would circulate throughout the body. Is that correct?
Yes, we tried different routes, right? Typically, fish oil taken by oral. The next speaker certainly will also emphasize that injection, acute treatment may be more effective, right? If you have acute inflammation, and either IV or sort of local tissue administration, or even to the spinal cord, the CSF. So these you need much lower concentrations, right?
Okay, well, thank you, Ru-Rong, and save your questions for the next session.